Hello, AP Bio, and this screencast is going to be about the humoral response. And the humoral response involves the B cells, and B cells are giving rise to plasma cells and memory cells, and the role the helper T cell plays. And further explore the why when you're exposed second time, so there's a quicker response and the basis of vaccines and AIDS. Now, again, we come back to this graphic organizer, and we went over cell-mediated response. So as you can see, first of all, you are exposed here to the antigen, and remember, antigen is something that elicits a response. It's a protein or polysaccharide from a pathogen, which could be a bacteria, a virus, and this is engulfed by macrophage, which will uh, digest it and present it on the cell, and that will stimulate the helper T cell. Now, the helper T cell will recognize it, and that will stimulate either a cell-mediated response, activating cytotoxic T cell and also memory T cells, or it will match up with a B cell and activate the B cells of this. B cell will then give rise to plasma cells, which are cells that will secrete antibodies, or put it in and record it into his memory B cells. So first of all, in our immune cells, we have the self versus non-self. So these self proteins, how does these cells know to recognize and distinguish between the self and non-self? And that is done by these proteins called MHCs, which stand for Major Histocompatibility Complex. And you'll hear MHC1 and MHC2. So they basically label the cell as self. And then what happens is when you have an antigen, that antigen is engulfed by a macrophage and then presented on these MAC markers, warning the antigen or advertising that this is the antigen. So, so here is an antigen that elicits the response, and it could be part of the pathogen that the antigen presenting cell will present, or it can directly be recognized by the cell. So the humoral response, or we also call it the adaptive immune response, is specific, and it responds to that particular pathogen. It's diverse. We have thousands and thousands of B cells and T cells that will have this lock and key model that recognizes that protein or polysaccharide. And first of all, it can distinguish between self from non-self. And it will have a immunological memory. It will respond faster when it's exposed to it later on. So for each T cell and B cell, there is a particular single antigen. Okay, so on these B cells and T cells, there's receptors, and these are receptors that's going to recognize that particular pathogen. So the humoral response, basically, you have a variety of B cells that have these antigen receptors that will recognize that particular antigen. And once that antigen is recognized, the B cell will bind to it, and it will wait for the helper T cell to activate it. And once it's activated, it will produce clone memory cells, and it will produce a group of cells called plasma cells, which will produce antibodies. And antibodies is a key thing in the humoral response. So, first of all, if you remember, the macrophage engulfs that pathogen, and once it engulfs it, the lysozyme will break it up and take that part of the antigen and present it on the MHC molecule. Then the helper T cell will recognize by its receptor, and once it's recognizes, it gets activated and then activates. It will either activate the B cell and it will activate the cytotoxic T cell. So why do we have so many types of B cells with different receptors? And that is due to the rearrangement of DNA. So in our stem cells, we have in the undifferentiated B cell possibilities of recombinations of these segments of DNA. And by our RNA processing, um, these genes can produce many types of receptors. Now, if you look down here, this Y-shaped looking receptor, you have regions that's called V, and this is the area where the receptor will be different. And then the regions that will be the same is what's called the C region or constant. So here, once the antigen presenting cell matches up with the helper T cell, and remember the T cell has a CD4 receptor, it binds, cytokines are released from the APC cell which activate the T cell, and then the T cell will release cytokines or activate cytotoxic T cells and B cells. And remember, cytokines are signals that elicit a response from these immune cells. So here is a more detailed diagram of how a helper T cell will activate a B cell. So again, the helper T cell is activated. It will bind to an APC cell, and here you have a MHC2 receptor that has an antigen presented on it. And the helper T cell will recognize it, bind to it. That will cause a signal from the activated helper T cell. So once it matches here with the B cell, it will release that signal. That signal will cause that B cell to differentiate two types of cell: plasma cells that will produce antibodies, and memory B cells that will record that pathogen in its memory. Okay. So the humoral response is all about recognizing the antigen and the antibody fitting with that antigen region, which we call the epitope. So epitope is a small region on the antigen where the antibody binds to it. And it's like this lock and key model. They fit like a puzzle. And how does the antibody attack the antigen? By several ways. And so these are different ways an antigen can neutralize or protect it from invading. One way is neutralization, where it will just bind to the antigen in several areas and neutralize and prevent it from infecting other cells. Or it'll cause that bacteria or virus to agglutinate, which means to group, and so enhances phagocytosis, or the precipitation of the soluble antigens, and again increasing or enhancing phagocytosis. And lastly, complement fixation, which will cause a series of reactions or signals to cause lesions or form pores, so the cell will lyse. Okay. Neutralization, opsonization, and complement system, again destroying the invader or the infected cell. So here this graph shows how when you're first exposed to the antigen, it takes time for the immune system to respond and form antibodies. And when it's exposed, so again, you see the time is between 7 to 28 days. But in the second response, it's much shorter. Okay. Now, HIV directly affects the T cells. So here, as you can see, when the body is exposed to HIV, you start forming those antibodies. So when we test for HIV, this is that we're testing to see if the antibodies are there. And as you can see, our body starts forming these antibodies, but then the HIV kind of takes over the T cell, inactivating it, and then HIV, the amount of HIV viruses increases. So again, coming back to this major graphic organizer, this is really important to understand, again, the humoral response versus a cell-mediated response. And knowing how the helper T cells is like the mediator, and it stimulates both the cell mediated response and the B cell. Now, allergies are overreactions, what means that our body recognizes things that are as invaders when it should not. So, for example, pollen is recognized as a pathogen, and that elicits in our cells called the mast cells that will release a lot of histamines, and these histamines will cause inflammatory response. So if it's in your nose, it's causing all this mucus to form, and you have this overreaction. So come with questions. Thank you for listening, and see you later.